Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the monosaccharides and disaccharides. Under the monosaccharides, we studied about ribose sugar, 2-deoxyribose sugar, and then under this hexoses separately, we studied about these two are ribose and deoxyribose are pentoses. Under the hexoses, we studied about glucose, galactose, mannose, and then even we studied about the fructose. And with that, we have studied about the monosaccharides. Then we studied about the disaccharides. Under the disaccharides, we studied about the sucrose, that is also called as invert sugar, and then we studied about the lactose, and then also we studied about the maltose. So these are the three very important examples we studied under disaccharides. After that, if 3 units are there, trisaccharides, 4 units are present, tetrasaccharide. Likewise, in the oligosaccharides, 2 to 10 units are present. If only single unit it is present, that is called as monosaccharide. Up to that we have studied. If many units are present, many means what? More than 10. If many units are present, then it is called as polysaccharide. So in this way, now we are talking about some polysaccharides. So in this, sometimes hundreds and sometimes even thousands of units are also present. But here in the NCRT, they are talking only about the 5 important examples. 5 important examples. First, I will write that complete list of 5 examples. And here we are classifying polysaccharides into different types. In one classification, we are calling them as homopolysaccharides and heteropolysaccharides. In another classification, we are classifying polysaccharides into structural polysaccharides and storage polysaccharides. Now I will write all this information so that first we will try to understand what are the examples given in NCRT and how exactly we are classifying them. With that, you will get 50% of the information, and individually we will talk about each one 2 to 3 points. That's it. With this lecture will be completed. So in NCRT, very few examples they give. If you at least remember that information, that is more than enough. Unnecessarily you don't need to go for extra information. Clear? And we are studying for different points given in the different chapters of NCRT. So ultimately we are covering the NCRT content. Clear? Now first let us write what is the list of these examples. For the convenience, I am writing like this. Observe carefully. First we are talking about the cellulose, and next we are talking about the chitin. Okay, and then we are writing about the starch, and then glycogen, and then we are writing about the inulin. It is not insulin, it is inulin. Spelling is very important. INULIN, inulin is a polysaccharide, but insulin, INSULIN, insulin is a protein. Please observe carefully. Insulin is a protein that is acting as a hormone, whereas inulin is a polysaccharide. So in this way, total 5 examples they gave in NCRT. And now how exactly we are classifying these 5? Whether these are homopolysaccharides or heteropolysaccharides. Please observe this. All the 5 are from the NCRT. These 5 examples, let us see each one, whether it is homopolysaccharide or heteropolysaccharide. Cellulose is homopolysaccharide. What is the meaning of homopolysaccharide? Already I told you what is the homo and hetero means. Homo means same. That means all the units are same. That means it is having single unit. The same unit is repeated for several number of times. Same unit is repeated for several number of times. So in this way, in the cellulose, a unit is present. I will tell you what is that unit separately. The same unit is repeated for several number of times. That's why it is homopolysaccharide. Suppose 2 or more types of units are present, then it is called heteropolysaccharide. But cellulose is homopolysaccharide. Then what about the chitin? Chitin is also homopolysaccharide. In many MCQ books, they are giving the wrong answer. They are giving chitin as heteropolysaccharide. That is wrong. Chitin is homopolysaccharide. Please observe carefully. Next one, starch. Starch is also homopolysaccharide. Glycogen is also homopolysaccharide. And inulin, this is also homopolysaccharide. So now you can clearly remember this. In NCRT, 5 examples they gave, and all these 5 examples are homopolysaccharides only. That means in the NCRT, in this chapter biomolecules, they have not given any example of heteropolysaccharide. So you can easily remember this. Okay, but in higher classes you will study about heteropolysaccharides. But anyhow, in the cell chapter I told you about some of the heteropolysaccharides. Now also in this chapter, here also at the end, just one or two sentences I will tell you. You need to remember that. Okay, but anyhow, this in this chapter NCERT content of 5 examples are examples for homopolysaccharide. Homopolysaccharide means it is having many units are present, but all these are exactly same units. So what is that monomer? What is that monomer? Individual unit. What is the individual unit in these cases that we are observing now? So what is the monomer in this case? Observe carefully. In the case of cellulose, we are saying that many units are present. Suppose this is one unit, second unit, third unit, likewise many are present. But we are saying that all the units are same. Then what is that individual unit? One unit. What is the one unit in the case of cellulose? Cellulose is having beta glucose. Beta glucose. See, generally students are remembering only glucose, but my suggestion is please remember whether it is alpha or beta. That may be useful in understanding some of the concepts. Okay, so please remember here in the case of cellulose, beta form of the glucose is present. How to write alpha form, beta form? I already told you. In that ring form, suppose you are writing like a linear one, then on the right side OH, on carbonyl carbon it is alpha. If left side OH it is beta. Suppose you are writing about this particular ring form like this, if you are using this pyranose ring form on this carbonyl carbon, suppose this is the first carbon, if OH is present above, it is beta form. If OH is present below, this is alpha form. Already we studied in previous lectures. Quickly revise that, you will get the information. So here what we are saying, it is beta form of the glucose. Next one, in the case of chitin, N-acetyl glucose amine is present. N-acetyl glucose amine is present. That means it is some complex molecule. Why it is complex molecule? It is not just glucose. Here what you are saying, to the glucose amine group, NH2 group is attached. To the glucose NH2 group is attached. And in this amino group N is there, no? To that N, acetyl group is attached. Please remember this. To the glucose amino group is attached. In that amino NH2, this N is there, no? So here it is H2. So you just remove one H. And then here to this N what happens? Acetyl group is attached here. So now what it is called as? So to the glucose amino group is attached. In this amino group to the N, acetyl group is attached. That's why it is called N-acetyl. That means acetyl group is attached to N of amino group attached to glucose, and it is called as N-acetyl glucose amine. Understood or not? So it is simply called as NaCGLU. That means N-acetyl glucose. Anyhow, this is amino group. So in this way, N-acetyl glucose amine. So this is what we are writing here. So in this way, here also it is the single unit. The single unit name itself is N-acetyl glucosamine. So in this way, many N-acetyl glucosamine units are attached to each other to form this chitin. Clear? Next one. In the case of starch, also, at least you know about this. Here also it is the glucose. But in the case of starch, it is alpha glucose. Next one is regarding the glycogen. In the case of glycogen, also alpha glucose. In the case of glycogen also it is alpha glucose. That's why glycogen is also called as animal starch. I will tell you all these points. First I am telling you the basic concept. If you understand basic concept, remaining things are just individual simple points that you can understand. See, actually starch is seen in the plants. But similar type of compound is present in the animals, and this is called as glycogen. So units are same, bonds are same, everything are same. Then why you are calling them with two different names? Because of the branching. That I will tell you separately in the individual explanation. Last one is regarding the inulin. What is this inulin? Inulin is also homopolysaccharide, but this is having beta fructose. Inulin is having beta fructose. Many beta fructose units are attached to each other. Understood or not? So this is how they are present. Okay, at least this is the basic information that you need to remember. So some extra information like what is the bond that is present. Beta 1,4 linkage is present in the case of cellulose. In the case of chitin, generally they will not ask this particular bond location and all, so you don't need to worry about this particular bond. Okay, next one, regarding the starch they will ask. In the case of starch, alpha 1,4 is seen and alpha 1,6 is also seen. Where it is seen I will tell you separately. Okay, next one, glycogen also same. Alpha 1,4 bonding and alpha 1,6 bonding is also seen. Okay, next one, in the case of inulin, here also they will not ask you the bond, it is actually beta 221. Beta 221 is the bond that is present. At least these three you please remember the bonds also. Okay, and this is what the basic information, and in this information what we wrote, whether they are homopolysaccharides or heteropolysaccharides, that information we wrote. All these 5 examples are homopolysaccharides. Cellulose is polymer of beta glucose, chitin is polymer of N-acetyl glucosamine, starch is polymer of alpha glucose, and glycogen is polymer of alpha glucose, inulin is polymer of beta fructose. So in this way, in all these things only single type of units are present. This is what about whether it is homopolysaccharide or heteropolysaccharide. Homopolysaccharide or heteropolysaccharide. In this way, we are writing the examples here. Clear? For heteropolysaccharides, what are the standard examples? You can talk about that hemicellulose pectins. Hemicellulose pectins, these names we studied in the cell chapter. When we studied about the cell wall structure, there we studied about this. Hemicellulose and pectins names we studied. There itself I told you, these are examples of heteropolysaccharides. Different units are present. But anyhow, these names we studied in cell chapter, and now you can interlink here. So hemicellulose and pectins are the most important examples for heteropolysaccharides. But in this chapter, whatever the 5 examples they gave, all the 5 examples are homopolysaccharides. This is one basic information you need to remember. Next classification we are studying. Polysaccharides are again classified into structural polysaccharides, storage polysaccharides. What is the significance of these words? You can understand this. Structural polysaccharide, that means these polysaccharides are making some structures. They are involved in formation of some structures. That is what the function is. Then what is the meaning of the storage? Some sugars are stored in the form of polysaccharides. Some sugars are stored in the form of polysaccharides. That's why they are called as storage polysaccharides. Now you need to understand this. Why a cell is storing sugars in the form of polysaccharide? Because later on these stored polysaccharides can be used as energy sources. Please understand this. These stored polysaccharides, whatever they are stored inside the cell, whenever there is a requirement, then cell will break that storage polysaccharide and then it will get the individual units and again it will get energy from that. That means the significance of storage polysaccharides is they are energy sources. They can be used as energy whenever it is required. In this way, 2 types are present. Understood or not? So now what we are doing is, whatever the 5 examples they have given in NCERT, for the same 5 we will write what type of polysaccharides are those. Cellulose, where we have studied about cellulose, let us go logically. Where we studied about the cellulose is present in the cell walls of the plant cells we studied. In the plant cells, cellulose is present. That means, is it storage or structural? It is making some structures called cell wall. So that's why cellulose is a structural polysaccharide. Cellulose is structural polysaccharide. It is helping in formation of some structures. Next one, chitin. Where we studied about the chitin. Chitin is present in the cell walls of the fungus. And then also we studied about, so this chitin is also present in the exoskeleton of the arthropods, some insects, yes or no? So we will study, slowly we will study about that cockroach also. In the case of the cockroach, we will study about the exoskeleton. Some chitinous plates are present. These plates are made up of the chitin. So there also they are making some structure. So that's why chitin is also structural polysaccharide. Chitin is also structural polysaccharide. So in this way, next one, starch. Where we are studying about the starch, starch is present in the plants. Where it is present? inside the cells. What we are saying, plant cells, they are obtaining the energy from the sunlight, so that they are using that sunlight energy, and this energy is used for the synthesis of some chemical compounds that is called as starch, and this entire process is called as a photosynthesis. So in the plant physiology there is a chapter called as photosynthesis. There you will study about this one. So plants are using the energy from the sunlight, and that energy they are using for the synthesis of some chemical compounds like starch. And what is the starch? It is a polymer of glucose. And why a plant cell is producing the starch? In future, whenever it is requiring energy, it should use from that particular one, and for that purpose it is synthesizing the starch. So the starch is ultimately used for energy source, and the starch is synthesized and stored inside the cell in the form of starch granules. Within the cytoplasm it is storing this one, yes or no? So in this way the starch is stored inside the cell. So that's why it is the storage polysaccharide. Starch is storage polysaccharide. Next one is regarding the glycogen. So in the case of animal cells, in the case of plant cells, this food materials are stored in the form of starch. But in the case of animals, it is in the form of a glycogen. In the form of glycogen, they are stored mainly in the liver and also in the skeletal muscles. This glycogen is stored. Slowly in those chapters separately we will study, so don't worry. So this glycogen is a storage, is also a storage polysaccharide in the case of animal cells. And next one is regarding the inulin. Separately they have mentioned in this one, and this is also a storage polysaccharide. Storage polysaccharide in the case of dahlia plant. This is also a plant. In the case of a dahlia plant, so it is stored in the form of inulin. Okay, so in this way, in the NCERT they have given the 5 examples. Out of these 5 examples, 2 examples, what are those 2? Cellulose and chitin are examples for structural polysaccharides. They are helping in the formation of some structures. And the 3 examples, starch, glycogen and inulin, are the examples of storage polysaccharides. In different organisms, some food materials are stored in these forms. Understood or not? In this way, polysaccharides are classified based on their functions into either structural or functional polysaccharides. Based on their structure, we are classifying them into homopolysaccharides and heteropolysaccharides. I hope now you understood this basic information. Now we need to talk about individual component. Once we complete that, this part will be completed. Clear? So this is what the basic information. So please observe it. So you don't need to do by hearting of this. Think logically so you can easily understand all these points. This is what about the polysaccharides. Out of this, first we are talking about the cellulose. So in the case of the plant cell walls, generally we are observing this cellulose. So the cellulose is having, see, very long threads. These are nothing but simply threads, like, so they are continuous long thread like filament like structures, right? And again they are made up of so many units. In different types of polysaccharides, different units are present. Now we are talking about the cellulose. In the case of cellulose, what is this? Each unit is called as glucose. Glucose. But what is the form exactly? It is the beta glucose. So it is the polymer of beta glucose. And it is a homopolymer. Because all the units are same, it is called homopolymer of beta glucose. And what is the bond that is linking this one here? Beta 1 to 4. One of this, 4 of this, one of this, 4 of this, one of this, 4 of this. That's why it is beta form 1 to 4 linkage. Understood or not? In this way, they are attached to each other. And this is the most abundant organic molecule. The sentence was clearly mentioned in NCERT. You need to compulsorily remember this. Cellulose is the most abundant organic molecule. If you are talking about individual components, the most abundant organic molecule is this cellulose. Okay, next one. And plant cell walls. It is generally seen in the cell walls of the plant cells. Next one. So this cellulose, using this cellulose, we are preparing the paper. See, in our textbooks or notebooks, whatever the paper that we are using are the newspapers. Whatever is the paper, this paper is mainly made up of plant pulp. Plant pulp means what is present in that? Again, it is mainly cellulose. So that's why this cellulose is the major one that is used for preparation of paper. And not only this, even the cotton. So in the case of cotton plant, that cotton fibers are produced. Yes or no? If you are technically and if you are chemically observing that cotton fiber is nothing but again cellulose. So cellulose fibers, cotton fiber is nothing but cellulose fibers. That's what they are saying. These are some examples given in NCERT. You need to remember this. And next one, in the case of cellulose, helices are not present. See, in the case of protein, we studied some helical structure, right? Likewise, for the cellulose, we are not observing helices. Why unnecessarily we are talking about helices here? Because we will talk about the helices for the starch molecule. Please observe this. In the case of starch, we are observing helical structures. Starch is showing helical structure so that it can hold iodine in between this and it will give blue color. Who is giving blue color? Starch is giving blue color. Why starch is giving blue color? Because it is having helical structure. Inside the helical structure, it is binding with the iodine. So that's why it is giving the blue color. But cellulose is not having the helices. So it cannot hold the iodine. It cannot hold the iodine. That's why it cannot give blue color. So generally this iodine is red or pinkish in color. So it will be like that only. So that means what they will do. Suppose you are taking the cellulose and you are taking the iodine solution and you are dropping the iodine solution here. Still it will be in the same color. Color of the iodine solution will not change because it is not having the helices and it cannot hold the iodine. This is what they are saying. But in the case of starch, you are taking the starch and you are adding the iodine. Now all of a sudden it will turn into blue color. Because this starch is having the helices, inside this helices it can hold the iodine so that it is giving the blue color. This is what the logical experimental thing, but sentences they gave in NCERT, you need to remember this. Okay, this is what about the cellulose. Very simple information, just 3 to 4 points they gave, that's it. Next one is regarding the starch. Starch is a storage polysaccharide in the case of the plants. So it is a homopolymer of alpha glucose units, already I told you. So many alpha glucose units are attached to each other. Okay. So here what happens? The starch is having the helical structure. Please remember these helices are also secondary structures. So you can understand this. DNA double helix is a secondary structure. Alpha helix of protein is also a secondary structure. And even the helices that are present in the starch are also secondary structures. All these are secondary structures. Okay, please remember this. And these helices will hold the iodine. So that's why when we are adding the iodine solution, now it will give the blue color. So this blue color is a characteristic feature. We can easily observe this presence of the starch with the iodine reaction. Okay, this is what they are saying. Okay, and generally if you are observing the starch, in the starch again two components are there. In some cases, some molecules are linearly present like a continuous, linear molecules. And in some cases, what happens? This is a line and here there is another one that is attaching. Now I will draw the rough diagram for that, observe carefully. So here what we are saying, here there are two components in the case of, starch. In the case of starch, two components are present. One is called amylose and other one is called as amylopectin. Other one is called as amylopectin. In the case of amylose, what happens? Suppose this is glucose. Likewise, linearly they are present. That's it. Here there is no branching. So it is unbranched. Unbranched form is called as amylose. Whereas in the case of amylopectin, what happens? So here there are some linear chains are present. So randomly I am drawing one rough diagram. Observe carefully. So this is one linear. And here somewhere, now this is one that is attached to this. Understood or not? Somewhere here there is a sequence that is attached to this. Understood or not? In this way they are attached. That means what happens? It is a linear, whereas this one is a branched one. Observed or not? So in this way, linear form is called as amylose, whereas branched form is called as amylopectin. Now you just observe the bonds carefully. Now in the case of amylose, what happens? Here they are all linear. So this is 1 and this is 4. This is 1, this is 4. 1, 4, 1, 4. So in this way, 1 of first one, 4th of next one. First 1 of this one and 4th of the next one. So in this way they are attached. So here also we are observing the same thing. 1 of this, 4 of this, 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 1 of this, 4 of this. So in this main line, 1, 4 linkage. Same here also. 1 of this, 4 of this, 1 of this, 4 of this, 1 of this, 4 of this. Here also 1 of this, 4 of this, 1, 2, 4, 1, 2, 4, 1, 2, 4, 1, 2, 4. This is what we are observing in the main chain. But wherever there is a branching, so this point is called as a branch point. This point is called as a branch point. So at the branch point, what is the bond? 1 of this and 6 of this. Why 6 of this? Because already this 4 is in bonding with this one. This 1 and this 4 are already in the bonding. So that's why what happens. This 1 is bonding with 6 of this. So that's why it is 1 to 6. 1 to 6 bonding is present only at the branch points. So that's why what is the branch point bonding? Branch point bonding in the case of starch or specifically in the case of amylopectin. What is this? It is 1,6. In the normal chain, what is the bonding it is? It is 1,4. That's why in the case of amylopectin, we are observing both 1,4 and 1,6 bonding. Understood or not? But in the case of amylose, it is only linear. So that's why always it is alpha 1,4 only. So overall, if you see both alpha 1,4 and alpha 1,6, that's what I wrote in that first slide. Yes or no? So in this first slide, this is what I have written here for the starch. This is what we have written. Understood or not? This is what the point is. So in this way, starch is having both linear and branched forms. Linear one is called amylose, branched one is called as amylopectin. Understood or not? Even in this one also, if they are specifically asking about in the starch, we are saying helical structures, right? Who is exactly having the helical structure? Suppose, is it only amylose or only amylopectin? It is only the amylose. It is the in specific if they ask. In general, if they ask, starch is having helical nature. That's why it can hold the iodine. That's why along with the iodine solution, it is giving the blue color. But in specific, if they ask, it is the amylose. It is the amylose that is having helical nature, so that it can hold the iodine, so that it is giving the blue color. This is what the basic information is. Simple logical points. Observe carefully, it will be completed. That's it. So in this way, in the case of amylose, only alpha 1,4 bond. But in the case of amylopectin, alpha 1,4, and at the branch points, alpha 1,6 bonding is also present. So in this way, at the branch point, one of the 6 of this is attached, because already this 4 is in bond with this one. This is what the logical point is. That's it. So in this way, we are studying about this starch. With that, information regarding starch is completed. Now next we are studying about the glycogen. How to remember the glycogen structure? Whatever the structure we are writing for amylopectin, exactly as it is same for glycogen also. That means here also it is having the branches. Here also it is having the branches. Then you may have the doubt. Then why you are giving a separate name called glycogen? Why can't you call this as amylopectin? Now there is a small difference, small technical difference. What is the small technical difference? More branched than starch. Here also you are observing the structure similar to amylopectin, but branching in the case of amylopectin is very less. Branching in the case of amylopectin is very less. But the branching in the case of glycogen is very extensive, very huge branching, extensive branching we are observing in the case of glycogen. This is the point that is making the difference between glycogen and amylopectin, or in general, glycogen and the starch. Here also alpha units. In the main line, straight line, it is showing the alpha 1,4. Only at the branching you are observing alpha 1,6. Bonds wise, it is exactly same. Units wise, it is exactly same. The major difference is with respect to branching. More branching is seen in the case of glycogen, less branching is seen in the case of starch, specifically amylopectin. This is the only difference, that's it. And here it is called as animal starch, because this structure is almost similar to starch of plants, but this is present in the animals. That's why it is called as animal starch. Glycogen is also called as animal starch. I hope now you understood this. And this is the storage molecule. So in the case of human, since we are animals, we are storing the food in the form of this glycogen. Yes or no? So whenever we need the energy, now this glycogen will be broken down, so that now it will start releasing the glucose. And from that glucose, we are getting the energy. That's what. Anyhow, there is a chapter called as respiration in plants. Even though you are say the chapter title is respiration in the plants, that glycolysis, Krebs cycle, electron transport, that basic concepts, they are applicable to both plants and animals. That concept is same for both plants and animals. But in NCERT, they gave that concept under plant physiology, that's it. But the reactions, everything are exactly same, even for animals also. So there you will study. So once glucose unit is released from this, it will be used for glycolysis, Krebs cycle, electron transport, so that we are getting energy. That's what they are saying here. So in this way, glycogen is one important example. And then that to glycogen is storage polysaccharide like starch. And next another one we are studying, this is inulin. Spelling is very important. It is inulin. It is not insulin. It is the polymer of fructose. That's why you can call it as a fructan. Okay. And here it is the beta fructose. It is the beta fructose. Bond is not important here. And where we are observing this? This we are observing in the roots of dahlia. I hope now you know about this dahlia flower, right? So actually these roots, they are having some tuber like, some bulged portions are present. In that actually it is storing this particular inulin. Clear? But here in some MCQ books, they are giving some extra information. What is that extra information? Inulin, when injected into our blood circulation, now it is going to the kidney for the filtration, right? So please remember, so this will be filtered. This will be filtered so that it will be released, and our body cannot utilize this particular inulin. Please understand this. Inulin is not used by our body, and also if we are eating starch, if we are eating the glycogen, we can digest it, but we cannot digest the cellulose. Human cannot digest the cellulose. Please remember this. Herbivorous animals, so they are eating the plants, so they are having this enzyme called cellulase. So they can digest the cellulose, but we are not producing cellulase enzyme, so we cannot digest the cellulose and we cannot digest the inulin, but we can digest starch and glycogen, but we cannot digest inulin and we cannot digest the cellulose. Clear? So this is one important point that we need to understand. And next one is regarding the chitin. So chitin, so this is also a structural polysaccharide like cellulose, but cellulose is present in the cell walls of plant cells, but chitin is present in the cell walls of fungal cells. Not only in the cell walls of fungal cells, it is also seen in the exoskeleton of arthropods like cockroach and many other insects. That's what they are saying here. Okay. And it is the second most abundant organic compound. Most abundant organic compound is cellulose. Second most abundant organic compound is chitin. You need to remember this. Clearly mentioned in NCERT, you need to remember this. Next one, it is the homopolymer of N-acetyl glucosamine. It is a homopolymer in many books. In many books they gave it is heteropolymer. That is wrong concept. It is a homopolymer because this same unit is repeated for several number of times. So this is how it is, a homopolymer of N-acetyl glucosamine, and it is also seen in the cell walls of arthropods and the, sorry, not cell walls of arthropods, cell wall of fungi and exoskeleton of arthropods. This is what. And then when we studied about the cell chapter, there we used these names, mannans. What are mannans? They are the polymers of mannose sugar. Mannans are polymers of mannose sugar. What are galactans? They are the polymers of galactose. So you can consider these as homopolymers. But hemicellulose and pectins, they are having different units. That's why hemicellulose and pectins are heteropolysaccharides. Clear? With this particular information is completed. But here they are talking about one right side and left side and reducing and non-reducing, and that concept they are talking. So I will tell you about that particular concept here. If we complete that, this concept will be completed. Come to this particular one. Let us use the same example here. In this example, let us observe carefully. This is the first carbon. This is the first carbon. This is the fourth carbon. First carbon, 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 fourth carbon is in the bonding. And here we are focusing on the first carbon because that is the carbonyl carbon. And here the first carbon is free. First carbon is free. Observe this concept carefully. In the previous lecture, we studied about reducing and non-reducing sugars. In the previous lecture, we studied about reducing and non-reducing sugars. Non-reducing sugars. Reducing sugar and non-reducing sugar. Which one is called as reducing sugar? Those sugars which have free carbonyl group. That means free aldehyde or free ketone. Yes or no? That means that should not be involved in the bonding. So those sugars which are having free carbonyl group, those sugars are called as reducing sugars. That means it should have free carbonyl group. Free carbonyl group. Then what about the non-reducing sugar? Those sugars which are not having free carbonyl groups, they are called as non-reducing sugar. What are the examples we studied for reducing sugars? All the monosaccharides are reducing sugars. In the case of disaccharides, we studied lactose and maltose as reducing. But under the non-reducing, we studied about the sucrose. Why we studied sucrose as non-reducing? Because the first carbon of glucose and the second carbon of fructose, they are the carbonyl carbons, and both of them are involved in the bonding. That's why they are not free in the case of sucrose. Since they are not free in the sucrose, it is a non-reducing sugar. This is what the concept for reducing and non-reducing sugar. But here they are talking about reducing and non-reducing end. Reducing and non-reducing end. Same logic we will use. The end that is having free carbonyl carbon, that is the reducing end. The end that is having the free carbonyl group is the reducing end. Whereas the end that is not having a free carbonyl group is the non-reducing end. Non-reducing end. Please observe carefully. This is what about the non-reducing end. Now we just see what is the standard way of representation. This is the left side. This is the left side and this is the right side. This is the left side and that is the right side. And what is the relation we are saying? Left side end is non-reducing end, whereas right side end is reducing end. Observe carefully. Left side end is non-reducing end, whereas right side end is reducing end. This is what the logical point is. Understood or not? With this ends concept is also completed. But now listen my question. You just take any polysaccharide. You just take any polysaccharide. In these polysaccharides, so here you are observing one reducing end, one non-reducing, sorry, this non-reducing end and this reducing end. Hence you are observing, okay. But listen my question. Can you consider a polysaccharide as reducing sugar or non-reducing sugar? Will you consider a polysaccharide as a reducing sugar or non-reducing sugar? Here this is having a free, that's why will you say it as a reducing sugar? First of all, you need to understand that the term sugar is not applicable or not used for polysaccharides. The term sugar is not used for polysaccharides. This is the first important point you need to remember with respect to the term sugars. Sugars are sweet. Sugars are sweet to taste. If you are tasting this, they should be sweet. They are only called as the sugars. Okay, not all the sweetened ones are sugars. Nowadays we are talking about some amino acids that are also sweet in nature, but I am saying that polysaccharides are not sweet in taste. Polysaccharides are not sweet in taste. Then who are sweet in taste? Mono and disaccharides. Monodisaccharides mainly, they are sweet in taste. That's why the term sugar, technically sugar means sweet in taste. So the term sugar technically used for mono and disaccharides, and the term sugar is not used for polysaccharides. This is one point. And next point, these polysaccharides are considered as non-reducing. They are not considered as reducing. Please understand this. Okay, so in higher classes you will study about all these things because they are based on some reactions. So now I don't want to explain all those, so please understand this. Even though it is having free carbonyl group at this particular end, still polysaccharide is considered as a non-reducing. That's it. Okay, and here this is what about the term. And polysaccharides are not considered as sugars because they are tasteless. They are tasteless. They are tasteless. Okay, so this is what the one important point that you need to remember. With this, entire information from the NCERT is completed. Clear? And now let us quickly go with NCERT reading part. So regarding the polysaccharides, they have given some small information. Let us quickly go through this. The acid insoluble pool, already we studied, no? So in the acid insoluble pool, 4 components are present. Proteins are present, nucleic acids are present, and then polysaccharides are present and lipids are present. Out of this, already we studied about proteins, nucleic acids and lipids we studied, and then one that is remaining is polysaccharides. And these we are observing under acid insoluble pool or retentate. In that we are observing this. The acid insoluble pellet has polysaccharides, means carbohydrates, as another class of macromolecules. Polysaccharides are true macromolecules because they are polymers. Polysaccharides are long chains of the sugars. So individual unit is called as a sugar because the individual monomer is a sugar. In this way, many sugars are attached to each other so that they are forming the polysaccharides. This is what they are saying. They are threads. Threads means what? Long molecules. That's why they are saying thread. Not only that one, literally even a cotton thread. Even a cotton thread is a polysaccharide because there is a cellulose polysaccharide. So then in this way, they are literally present as threads containing different monosaccharides as building blocks. In different types of polysaccharides, different types of monomers are present. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. In the case of cellulose, only one type of unit is repeated for several number of times. And what is that unit? It is the glucose. That means cellulose is homopolymer of glucose. Next one, they have given. So cellulose is a homopolymer of glucose. Next one, with this cellulose is over. Now starch. Starch is a variant of this but present as a storehouse of energy in plant tissues. Why they are saying variant? Because starch is also having cellulose, but the form, if you see, starch is also made up of glucose, but if you are observing, cellulose has a beta form of glucose, but starch is having alpha form. That's why it is a small variant of this, but it is present as storehouse of energy in plant tissues. Why they are saying like this? Because cellulose is a structural polysaccharide, whereas starch is storage polysaccharide. It is storage polysaccharide because it will provide the energy. So that's why as storehouse of energy in the plant tissues. It is the storage polysaccharide in the plants. Next one. Animals have another variant that is called as glycogen. In the case of animals, starch is not present, but glycogen is the storage polysaccharide. This it is over. Next one. Inulin is a polymer of fructose. So inulin is a storage polysaccharide in the case of roots of dahlia, and it is made up of beta fructose. This is what they are saying. Next one. In a polysaccharide chain, let us say glycogen, anything you can take. In a polysaccharide chain, the right side end is called as reducing end, and the left side end is called as non-reducing end. Right side is reducing end, left side is non-reducing end. Reason also I told you. Okay, because on the right side end, a free carbonyl carbon is there, but on the left side, it is already involved in the bonding. It is not free. That's why they are saying like this. And it has branches as shown in the form of a cartoon. This is the cartoon they give. So it is the main linear one, and again it is having so many branches like this. Okay, so in this way, at the branch point, it is having the 1,6 linkage, but remaining all they are having 1,4 linkage. So in this way, so it is present. Next one. Starch forms helical secondary structures. Only the starch. Out of all this, starch is forming some helical secondary structures. In fact, starch can hold iodine molecules in the helical portion. In between these helices, iodine can be kept. So in this way, it is holding the iodine. That's why the starch iodine complex is blue in color. That's why when starch is interacting with the iodine, so it is giving the blue color. Originally iodine is having some reddish to pinkish color. Okay, this is what. Next one. Cellulose does not contain the complex helices and hence it cannot hold iodine. That's why cellulose cannot give blue color. Iodine, so starch can give blue color with iodine. That's what they are saying. This is the diagram that they have given. So this is the left side end. Left side end is non-reducing end. This is the right side end. Right side end is reducing end. That's what they are saying. Clear? Next paragraph. Plant cell walls are made up of cellulose. So cellulose is the structural polysaccharide present in the cell walls of the plants. Next one, paper made from the plant pulp. So that is made up of cellulose. That's what they are saying. Paper made up of the plant pulp and cotton fibers. All these are cellulosic. They are made up of cellulose. So cotton fiber is made up of cellulose, and paper that is made from this particular plant pulp, that is also made up of mainly cellulose. And there are more. Up to this we are talking about simple single unit and repeated for several times, and that too it may be direct glucose or fructose likewise. But now we are talking about some complex polysaccharide. Complex in the sense that each unit is not a normal glucose or normal fructose. It is again further modified, like N-acetyl glucosamine, N-acetyl galactosamine or muramic acid. Likewise, different things are there. Okay, in this way they are saying paper made from the plant pulp and cotton fiber are cellulosic. Over. Next one. There are more complex polysaccharides in nature. Not only this, in the nature, so many different other types of even complex polysaccharides are also present. And they have as building blocks. As building blocks, what they have? Amino sugars. That means to the sugar amino group is attached. In this way also, some sugars are present. And chemically modified sugars. Chemically modified sugars like glucosamine, to the glucose amino group is attached. N-acetyl galactosamine, to the galactose amino group is attached. In that amino group, to the N-acetyl group is attached. So in this way, some complex molecules are also present in the nature. In this way, they are also present. This is what they are saying. Next one. Exoskeletons of arthropods, for example, have complex polysaccharide called chitin. Why chitin they are calling as complex? Please remember, homo hetero is different and complex is different concept. Homo means same unit repeated for several times. So chitin is homopolysaccharide because N-acetyl glucosamine is repeated for several times. But what is the meaning of this complex? Why students are getting confused here? Here complex because chitin is not having just a direct glucose. Chitin is not having only the glucose. N-acetyl glucosamine, to the glucose amino group. In that amino again acetyl is attached. So that's why it is complex. But entirely it is one unit. Since same unit is repeated, it is compulsorily homopolysaccharide. It is complex because it is not directly glucose, it is actually a modified sugar. That's why it is complex. But why it is homopolysaccharide? Though it is complex, the same unit is repeated. That's why it is homopolysaccharide. So please observe carefully here. You should know the meaning of each and every word. Okay. So that's why chitin is a homopolysaccharide and is a complex polysaccharide. Understood or not? Next one. And these complex polysaccharides are mostly homopolymers. Chitin homopolymer. This is what they are saying. Even though they are complex, they are modified forms of sugars, but generally we are observing homopolysaccharides. So in this way, whatever the examples they gave in NCERT, all these examples are homopolysaccharides only. That hemicellulose pectin, these are some important examples for heteropolysaccharides having different types of units. Clear? With this, actually this lecture is completed, but before closing this, I will give you one summarized information. Observe carefully here. This is the left side. This is the right side. This information is summarized information. Every student should remember this compulsorily for examination. If we are talking about the DNA, what is the direction of DNA synthesis? Regarding DNA and RNA direction, that is called 5' to 3'. If you already know, that is very good. Remember this. But I will explain about this in the molecular base of inheritance. So please remember this. DNA synthesis, as per convention. What is our convention? There are some group of scientists, so they have decided, they met together and they have decided that we should use this particular terminology always. So that we are considering as the convention. What is that convention here? So left side, what is the end that we need to use? Right side end, what is the end? See here, when we are writing like this is left, this is right. So in that sense only we are writing. DNA synthesis, this is how we are writing. Left to right we are writing. So what is this left side end? For DNA, left side end is 5' end, right side end is 3' end. Please remember this, very very important for examination. For the DNA, left side end is 5' end, right side end is 3' end. Clear? Next one, for RNA. For RNA also it is the same. Left side end is 5' end, right side end is 3' end. For both nucleic acids it is exactly same, because these directions it is decided based on their synthesis. During synthesis, how exactly it is happening? DNA synthesis occurs from 5' to 3'. That's why we are writing like this. RNA synthesis occurs from 5' to 3'. That's why we are writing like this. But protein synthesis occurs from N terminus to C terminus. Protein synthesis occurs from N terminus to C terminus. That's why left side end for protein is N terminus, right side end for protein is C terminus. Next one for polysaccharides. For polysaccharide, what is this left side end? Just now we studied. Left side end is non-reducing end. Left side end is non-reducing end. and right side end is reducing end. And my suggestion is better you please pause this particular one and then write this particular information in textbook itself. In biomolecule somewhere you just write it. Okay, so this is what the information is. So what is the left side and represents, what is the right side and represents? As per convention you need to talk all this. Okay, as per convention you need to talk all this. As per convention you need to talk all these things. Is it clear? So in this way we are using these ends also. So with this even the polysaccharides information is also completed, and with this all the 4 components of macromolecules we studied, and along with this even some of the micromolecules like individual amino acids we studied, individual monosaccharide disaccharides also we studied, and individual nucleotides also we studied. So in this way all the components of that acid insoluble pole and some important components of acid soluble pole we have completed. Clear? With this these individual concepts are completed. So in the next lecture we can go with some other concepts. Okay, thank you so much my dear friends. We will meet in the next lecture. Thank you.